Hello everyone, and this is Dr. Rajni Sharma. I welcome you in my channel Be Yourself. Today I will discuss about gene, the structural and functional unit of life. Gene is a segment of DNA which consists of exons as well as introns. Introns are generally present in case of eukaryotes. It is a non-coding region, whereas exon is the coding region. Actually, DNA is a macromolecule which is present in the cell and generally packed into a structure called chromosomes. DNA also consists of highly repetitive or simple sequences, also known as satellite DNA. It is named so as it is involved in migration. This doesn't code the protein, rather associated with two important structures of DNA, which is centromere and telomeres. Centromere is AT-rich region and are involved in binary fusion and help in the equal distribution of chromosomes. Centromeres are the sequence that are at the end of eukaryotic chromosomes. It helps to stabilize chromosome. In case of yeast cells, telomeres has a repetition of TZ unit. Here X and Y represent the number 1 to 4 and represent the repetition of telomeres, which can be 20 to 100 times. The most critical thing about DNA for life is its packaging and segregation to daughter cells. In case of prokaryotes, its packaging is simpler as compared to eukaryotes. In case of viruses, less genetic information are present in their DNA as they fulfill their need from the host cells. For example, HIV has 9,000 nucleotides in length, whereas bacteriophage QB has 4,220 nucleotides in length. Both has single-stranded RNA genome. Bacteria has more DNA than virus. DNA of E. coli has 100 times more DNA than present in bacteriophage lambda. In addition to bacterial DNA in bacterial cells, it also contains plasmids. It also provides as a resistance to the bacterial cells. For example, it can provide resistance against certain antibiotics. Eukaryotic cells contain even more DNA than prokaryotes. Even organelles of eukaryotes, which include chloroplast and mitochondria, also contain DNA. The most important aspect of cellular DNA is their structural organization. They are highly compacted but do not affect the access of information in the DNA. To clarify this, we must have a glance over the supercoiling of DNA. Supercoiling is an intrinsic property of DNA tertiary structure. Actually, in DNA supercoiling, if it is in relaxed state, if we consider the case of prokaryotes which has a circular DNA, unless it will underwind, it will not change in the supercoiled form. As here I have relaxed DNA, when it will underwind, it will change to supercoiled state. Underwinding changes the linking number, which is a topological property. A number of measurable properties of supercoiling is studied under the branch of mathematics called topology. It deals with the properties of an object that do not change under continuous deformations. Deformations may be continuous, which include the changes due to thermal motion or an interaction with the proteins or other molecules, and this continuous deformation due to DNA strand break. Here I have two figures in which one have linkage number 1 and other has linkage number of 6. Now how this come? A linkage number is the number which says how much time the second strand has passed over the first strand. As here in figure 1 it has linkage number 1 and the DNA strand 1 has passed over the other strand and single time, while in the other figure it has passed 6 times over the first strand. When it is left-handed, then the linkage number is negative, and when it is right-handed, then linkage number is positive. In case of relaxed DNA, linkage number can be defined as base pair per base pair turn. Linkage number in supercoiled DNA is called the specific linking difference or superhelical density. It is equal to change in linking number by the linking number in relaxed state. If a relaxed DNA differs only in their linkage number, as here the linkage 2 figure a positive coiling, supercoiling and negative supercoiling differs only with the linkage number of -2 and +2. Such condition is known as topoisomerase. This number can be broken down into twist and wreath, or we can say that linking number is the sum up of twist and wreath types of folding of DNA. Topoisomerase are classified into two classes. Type 1 break one strand of DNA and rotate it into two unbroken strands. They change the linkage number in increment of 1, and type 2 breaks both strands of DNA and change the linking number in increment of 2. In case of E. coli, type 1 topoisomerase are of two types, type 1 and type 3, and these generally relax DNA by removing negative supercoiling, whereas type 2 Organelles introduce negative supercoiling and it uses ATP. Topoisomerase in case of eukaryotes include two types of type 1 topoisomerase, which is 1 and 3, just like that of E. coli, and type 2 has type 2 alpha and type 2 beta. This relaxes both positive and negative supercoiling. Now how it works. In case of relaxed circular DNA, when topoisomerase acts on a point, this is a condition, especially that of virus, it will lead to positive supercoiling. To compensate it, at another point there will be negative supercoiling. Then, at the place of positive supercoiling, the strand will break and undergo underwinding to change the linking number by -2. To For the compactness of packaging, there are generally two forms of supercoiling, plactonemic and solenoid form of supercoiling. You can clearly see here the difference between these two structures. Plactonemic and solenoid are interconvertible. Plactonemic form is more stable than solenoid one. Solenoid are stabilized by binding of proteins. This form is found in chromatin. DNA in chromatin is tightly associated with histone protein, which package and order the DNA into a structural unit called nucleosomes. Histones are small basic proteins which is rich in arginine and lysine, and these provide positive charge to histones. 5 major classes of histone proteins are present in eukaryotes. Histone 3 nearly identical in amino acids in all eukaryotes. Histone 4 strict conserved in their functions. H1, H2A and H2B are less sequence similarity among eukaryotes. Each of the histones has variant form because certain amino acid side chains are enzymatically modified by methylation, ADP ribosylation, phosphorylation, or acetylation. Such modifications affect the net electric charge, shape, and other properties of histones, as well as the structural and functional properties of chromatin, and they play a role in the regulation of transcription. In a unit of nucleosomes, there are the dimer of histone proteins. The dimer includes S3, H4, S2A, and S2B, over which 200 base pairs of DNA are arranged, but only 146 base pairs of DNA are around the nucleosomes, while the rest are involved in linking with the other unit of nucleosomes, and this is stabilized by histone protein H1, that is the which links the two nucleosomes in a single unit. That's all how DNA get arranged in the our cells in a very compact nature without disturbing its coding regions. Hope it will be very interesting videos about DNA. I will very soon upload new videos of DNA explaining their functions, explaining their metabolisms. So for that videos, don't forget to subscribe and share my channel. Be yourself. Till then, goodbye.